In this lecture, we are going to talk about integumentary system. I'm going to divide this system into 3 lectures. The first one, we will talk about epidermis. Lecture 2, dermo-epidermal junction, dermis and hair. Lecture 3 about sweat glands, nail, blood supply, functions of the skin, and effect of aging on the skin. First, what is meant by integumentary system? The word integumentary meaning that something that cover. Integumentary system is formed of skin and skin appendages, which are nails, hair follicles, sebaceous gland, and sweat gland. Skin is considered the largest organ in the body. It constitutes 16% of the total body weight. It consists of 2 layers, epidermis, which is the outer layer, and it is ectodermal in origin, and the dermis, which is mesodermal in origin, and it is the deeper layer. The skin rests on subcutaneous connective tissue, which is called hypodermis. It is equivalent to subcutaneous fascia. It is rich in fat. Notice that hypodermis is not considered as a part of the skin. According to the thickness of the epidermis, we have 2 types of skin. Thick skin, which is present only in the sole of the hand and palm of the hand and sole of the foot, and it is more subjected to abrasion and friction. And the second type is thin skin. The epidermis, which is a superficial layer of the skin formed of stratified squamous keratinized epithelium, which is ectodermal in origin, and it is separated from the deeper dermis by a well-developed basement membrane. As we see here, the dermis reveal dermal papillae, and the epidermis reveal epidermal ridges. So both of the dermal papillae and the epidermal ridges help in the fixation of the both layer epidermis and the dermis together. Regarding the structure of the epidermis, as we said, it is formed of stratified squamous keratinized epithelium, and this epithelium formed of 2 types of cell, keratinocytes, which represent 80% of the epidermal cell, and non-keratinocyte. Non-keratinocyte formed of 3 cells, as we see here, those cells, melanocyte, Merkel cell, and Langerhans cell. So we have, in other words, we have 4 cells in the epidermis: keratinocyte, melanocyte, Merkel cell, and Langerhans cell. Keratinocytes are arranged in 5 layers. The first layer is stratum basal, which is resting on the basement membrane. The second layer is stratum spinosum, and the third layer stratum granulosum, then stratum lucidum, and lastly stratum corneum. Again, in summary, integumentary system is formed of skin and skin appendages. Skin appendage is formed of nail, hair follicles, sebaceous gland, and sweat glands. Regarding the skin, it's formed of superficial layer, which is epidermis, and dermis, which is the deep layer. Epidermis is formed of keratinocyte or non-keratinocyte. Keratinocyte arranged in 5 layers, as we see here: stratum basal, stratum spinosum, stratum granulosum, lucidum, and corneum. However, the non-keratinocyte, as we said, 3 cells: Langerhans cell, melanocyte, and Merkel cell. Regarding the dermis, is divided into 2 layers: papillary dermis and reticular dermis. The first layer of the epidermis is stratum basal. By light microscope, it consists only of a single row of columnar cells, as we see here, which is resting on the basement membrane. The nucleus is appearing a single basal oval nucleus, and the cytoplasm is deeply basophilic and contains melanin granules. By electron microscope, it shows ribosomes, free mitochondria, rough endoplasmic reticulum, and small Golgi apparatus. The most important finding by electron microscope is ribosomes. Why? Because stratum basal is responsible for the regeneration of the other layers of epidermis. So it reveals a lot of or plenty of ribosomes. It also contains intermediate filaments, which is tonofilament or keratin, another name keratin filament or cytokeratin filament. Cells of stratum basal attached to the basement membrane by hemidesmosomes and attached to each other by desmosomes. As we said, it show high mitotic activity. It is considered as a stem cell so that it contains plenty of ribosomes. In this layer, stratum basal, two cells also present, Merkel cell and melanocyte. Both of those cells, melanocyte and Merkel cell, present in this layer, stratum basal. It formed of multiple layers of polygonal cells with a single central rounded vesicular nucleus. These cells are larger near the basal layer than near the surface. They have spiny appearance. In this layer, which cell of the non-keratinocyte present? Langerhans cell. So in the stratum basal, in addition to the keratinocyte, there are another two types of cells, which is melanocyte and Merkel cell. The two cells starting by letter M, and here in the stratum spinosum, in addition to the keratinocyte, also Langerhans cell is present. In this layer, stratum spinosum, the keratinocytes are attached by desmosomes. Which are located at the tips, as we see here, at the tips of the spiny projection. The desmosome present at the tips of the spiny projection of the keratinocyte to give good attachment between the cells. As we said, skin is subjected to the outer surface and subjected severely to the friction. A general rule, as we direct superficially in the layers of the epidermis, there are more tonofilament or more keratin filament, and as we going superficially, melanin granules decrease. So the melanin decrease and the tonofilament or keratin filament increase. Cells of stratum spinosum are also mitotically active, and both stratum basal and stratum spinosum called stratum germinativum. So that what is meant by stratum germinativum? It means both stratum basal and stratum spinosum. It means the cells which have the ability to regenerate the other cells of epidermis. And the regeneration of the epidermis is completed about every 15 to 30 days. The third layer of epidermis, stratum granulosum, or the other name, granular cell layer. By light microscope, it formed of 3 to 5 layers of flattened cells. Here in this layer, the cell started to be flattened with central oval nuclei. In this layer, by electron microscope, the cytoplasm contains, as we said, keratin filament. As we said, as we going superficially in the epidermis, the keratin filament increase and the melanin granules decrease. In addition to the tonofilament, there are two types of granules, so that its name is stratum granulosum. The first type of the granules is keratohyaline granules. They appear coarse basophilic, irregular granules. They are not surrounded by membrane. Its function it contains protein that bind the keratin filament together to make a more denser or a more thicker bundles. The second type is of the granules is membrane coating granules, which is granules surrounded by membrane and called lamellar granules. It appears electron dense by electron microscope. The second type, which is the lamellar granules, it arises from the Golgi complex, then excreted to outside of the cell by exocytosis, discharged outside the cell in the intercellular space and forming, as we see here, a membrane, intercellular, as we see here, layers between the cells in the form of lipid lamellae, which is responsible for the water barrier or waterproofing of the skin, and it prevents the water loss from the skin. It also acts as a barrier against the foreign substance or the bacteria, and in addition it acts as a cement substance to connect the cells together. Layer number 4 of the epidermis is a stratum lucidum, which is a clear cell layer. It is a homogeneous translucent layer, and take care or notice that it is only present in the thick skin, not present in the thin skin. It is formed of flattened acidophilic cell, and in this layer the cell starts to lose its nuclei and organelles. In the stratum lucidum, the keratinocyte start to densely packed with keratin filament, which is embedded in electron dense matrix, and they are attached by desmosomes. Layer number 5, which is the last layer, is a stratum corneum or horny cell layer. It is the most superficial layer and formed about from 16 to 20 layers. This formed of several layers of non-nucleated keratinized or fully keratinized acidophilic cells. There is no nuclei, there is no organelles, the cytoplasm only full of keratin tonofilament, and the cell appear dead keratinized cells embedded in amorphous matrix. By time, the cells of stratum corneum shed off continuously from the surface, and it is replaced by newer cell from the deeper layer, which, as we said previously, stratum germinativum, which is stratum basal, and stratum spinosum. Here in this layer, the cells are adherent to each other by remnant of desmosomes. As we see here, this is the first layer, stratum basal, which is columnar cell resting on the basement membrane. It contains tonofilament and melanin granules, and it is attached to the basement membrane by hemidesmosomes and attached to each other by desmosomes. The second layer is a stratum spinosum, which contains tonofilament. And as I said, as we directed superficially, the tonofilament increased. After that, stratum granulosum, which contain two types of granules, either keratohyaline granules, which is not surrounded by membrane, and these granules contain protein to bind the tonofilament or keratin filament together to form bundles. And the second type of granules is lamellar granules, which discharged into the intercellular space by exocytosis to form a lipid layer or waterproofing to protect the skin from the water loss. After that, stratum lucidum, which is only present in the thick skin. And lastly is a stratum corneum, which is fully keratinized cells devoid of nuclei and in this issue, I would like to talk about psoriasis. Psoriasis is a chronic اللي هو بالعربي الصدفية. It is a chronic autoimmune disease characterized by abnormal increase in the proliferation rate and the differentiation of the keratinocyte, so that there is imbalance between the newly formed cell, which exceed the regenerated cell. As we said, the normal rate of skin renewal from 15 days to 30 days. This imbalance in the rate between regeneration and desquamation lead to epidermal increase in epidermal thickness, increase in desquamation and scaling, and inflammation due to defective skin barrier. Also different type of skin, malignant skin tumors arise from the cells of the epidermis. We have basal cell carcinoma, which arise from the basal cell layer. We have a squamous cell carcinoma, which arise from the stratum spinosum or spinous cell layer, and we have malignant melanoma, which arise from the melanocyte. We have also blistering disorders of the skin. We have friction blisters or callus. It is formed of, or it is in the form of lymph-filled spaces between the epidermis and dermis of the thick skin. Occur in the thick skin due to excessive wrapping or friction. It is occur to all of us when we wear a new shoes. The second type is blistering due to autoimmune damage or damage to the intercellular junction between the keratinocyte. It leads to a disease called pemphigus. After we talk about melanocyte, the second cell we are going to talk about is melanocyte, which is a melanin forming cells. It is present, as we said, in the stratum basal layer and also present in the hair follicles to give the color of the hair. It is ectodermal in origin, arise from the neural crest. By light microscope, it is a branched cell and it has long processes extend between, as we see here, extend between the keratinocyte. This is the melanocyte, has rounded cell body and central vesicular rounded nucleus. As tyrosinase enzyme act on tyrosine, leading to the formation of DOPA, so that when we examine the presence of DOPA in melanocyte, it reveals positive reaction. Tyrosinase enzyme formed as it is a protein. It is synthesized in the rough endoplasmic reticulum and then accumulated or packaged in vesicles by Golgi apparatus. Tyrosinase enzyme, as I said, convert tyrosine into DOPA, which converted after that to melanin pigment. As I said before, a melanocyte present in the stratum basal resting on the basement membrane and attached to the basement membrane by hemidesmosomes, and its processes extend between the stratum spinosum layer. As it forms the melanin pigment or melanin granules, it injects it into the keratinocyte by cytokine secretion. Here is the keratinocyte. Here is the keratinocyte of the stratum basal, and as we see here is the arrow the melanin granules, which is supranuclear to protect the nucleus and to protect the DNA from the mutation effect by the ultraviolet rays. What is the function of melanocytes? Melanocyte responsible for the skin pigmentation. It gives us our color. The second function of melanocyte, it protects us against the ultraviolet ray, harmful effect of the ultraviolet rays, as these rays affect or leading to mutation or mutating effect in the DNA. So it protects us from these harmful rays. Here is a table to differentiate between melanocyte, which is just talking about it now, and the melanophores. What is melanophores? It is one of the connective tissue cell. I think you studied it in the first term in the chapter of the connective tissue. It is other name is pigment cell. It is mesodermal in origin, and it phagocytoses the melanin formed by melanocyte. Is it DOPA positive or negative? It has no tyrosinase enzyme, so that it is DOPA negative. However, on the other side, melanocyte contain tyrosinase enzyme, so that it is DOPA positive. Here we are the page all of us wish to be there now. in these conditions. Type. What is the effects of sun rays on the skin? How the sun rays affect our skin? It lead to darkening of our skin, or in other words, tanning. How it gone? Darkening of the pre-existing melanin. The pre-existing melanin become more dark, and in addition to that, there is increase in the synthesis of melanin due to paracrine factors. In addition, exposure, a lot of exposure to sun rays lead to destruction of the elastic fibers. So please all avoid to expose to the sun rays for a long time, except you are putting sun protector or sunblock. And now, what is the difference between the dark skin and light skin? Why some of us have dark skin, or why we reveal different degrees of color of the skin? Is it due to the number of melanocytes? No, as we reveal here, or as I show here, the number of melanocytes is constant. Here we have one. In the dark skin, this is the dark skin, and this is the light skin. In the dark skin we have, for example, one melanocyte, and in the light skin we have also one melanocyte. The difference between both of them, as we see here, the slow or the rate of degradation of melanocyte by lysosome as we go upward. In the dark skin, there is slow degradation of the melanocyte or melanosome as we go upward. However, in the light skin, there is a rapid or a high rate of degradation of melanosomes or melanin pigment as we directed upward. And this is the cause of variation in the color of the skin between us. Here we have two abnormalities in the skin pigmentation. The first one is albinism. It is a congenital disease. In this disease, there is hypopigmentation of the skin due to defect in the tyrosinase enzyme or any steps in the pathway of the melanin formation. However, the vitiligo is an acquired disease. It is an acquired disease leading to depigmentation of skin in some areas due to also defect in the melanin synthesis or loss or decrease in activity of the melanocyte. The melanocyte start to show decrease in its activity, leading to depigmentation of some areas in the body. Again, as I said before, epidermis is formed of keratinocyte and non-keratinocyte. Non-keratinocyte, the first one is melanocyte. I already talked about it. The second is Merkel cell, and the third is Langerhans cell. Merkel cell is present in the stratum basal, as we said before. It is more numerous in the thick skin. It is especially in the fingerprints, as it is responsible for the fine touch. By light microscope, its shape is branching cell attaching to the neighboring keratinocyte by desmosomes, as we see here, and Merkel cell act as mechanoreceptor. It is responsible for the touch sensation. Merkel cell, as we said, is responsible for the touch sensation. They are sensory sense or touch receptor. In the underlying dermis, they receive afferent nerve. Which lose its myelin sheath and penetrate the basement membrane. Penetrate, as we see here, the basement membrane. This is the nerve. Penetrate the basement membrane and form an expanded terminal disc. It is called Merkel corpuscle or Merkel's tactile ending. So, what are the receptors present in the epidermis? We have two types of receptors. either the free nerve endings, which is responsible for pain and temperature, and the second one is Merkel nerve disc, which is responsible for the touch or fine touch sensation. The third cell of non-keratinocyte is the Langerhans cell, which originates from the bone marrow. It is present in the stratum spinosum. It is a star-shaped cells, as we see here. This is the blue one in the Langerhans cell. The star-shaped cells and have long processes extending between the keratinocyte, lacking desmosomes with them. Langerhans cell is not attached by desmosome to the neighboring keratinocyte. Langerhans cells contain, by electron microscope, contain numerous lysosomes and a characteristic racket-shaped granules called Pyre-Peak granules. These granules are membrane-bound granules, but unfortunately it is of unknown function until now. The function of Langerhans cell, it acts as antigen-presenting cell, play a role in the immune system. It represents the antigen to the nearby T lymphocytes. As immune I wish I can explain this lecture well, and I'll see you in the next lecture, inshallah.